Polak. He's a deep learning engineer at Polish-Japanese Academy of Information Technology, and he will hold the beginner's guide to MRI, brain scans, classification using machine learning with FastSurfer, XGBoost, and 3D CNN tutorials. Enjoy.
Thank you. Hello, everyone. Welcome to my tutorial. My name is Damian, and I'm a machine learning engineer from Poland. Most of my work revolves around computer vision and deep learning. Today, I will be talking about working with MRI brain scans using machine learning techniques. This tutorial is based on my experience gained while working on my master's thesis, which focused on the automatic classification of MRI scans for Alzheimer's disease prediction. The entire project for my master's took about 1 year of work, and I'm really grateful for the opportunity to share this knowledge here with you at the Data Science Conference. Okay, to get started, you will need to follow these steps. First of all, if you want to go through all the steps, including data preprocessing, you will need to install Docker on your machines. However, this step is optional. I've already uploaded the AXI dataset preprocessed by me to Dropbox. So if you don't have Docker installed or you don't own an NVIDIA GPU, you can skip this whole step. All the required code is included in the GitHub repository, allowing you to reproduce the preprocessing steps later on your own. If you're skipping the preprocessing step, you also don't need to download the original AXI dataset. Only the spreadsheet containing demographic data is required. The entire preprocessing process is time-consuming and takes hours, so I will only show you how to do it and how it works, using only one example. However, as mentioned earlier, you can download the preprocessed data. Additionally, we will need a FastSurfer license, which is free, but registration is necessary. Here are the links for the preprocessed dataset, original dataset, and tutorials repository. The first link is shortened URL pointing to Dropbox. Now let's proceed to download the data. You need to check this checkbox out and click download here. And now wait a minute. Okay, it started downloading. And here, as we are now downloading the dataset, we also need demographic tabular data from the AXI project site. And you can download the spreadsheet here. If you prefer to preprocess all the data on your own, you can also download the images from this site. Remember to choose T1 images if you want to precisely follow the tutorial, but feel free to experiment with other sequences as well. I will discuss what sequences are later. The third link is for the tutorials repo. Remember to download submodules as well. This repo uses my implementation of so-called Pseudo-3D convnets, which is stored in a different repo and is linked as a submodule. I will talk about these Pseudo-3D models later. And here's a single command to clone the entire repository. I can copy and paste it here for you. Okay, moving to the next slide. Here is a link to the form where you can obtain the FastSurfer license. And I will provide more details about FastSurfer later. And as mentioned before, these two steps are optional because the preprocessed dataset is already available for download. However, for your own projects involving other MRI brain datasets, FastSurfer will likely be necessary. So let's proceed with registering. On the FastSurfer site, you need to fill this form, pass the CAPTCHA test, and agree for the license agreement. You will then receive a license file to your mail. And assuming you have already installed Docker, as I have, and some of you may have as well, I will skip this step to save time. But as I said before, it isn't necessary. Maybe do you have any questions so far? We can take 1 minute break to address your questions, and you can use this time to download all the necessary files. Okay, so if you don't have any questions, let's delve into a short theoretical part. In this tutorial, I will guide you on predicting a subject's biological sex and age using machine learning techniques with MRI brain scans. Predicting demographic information may not be particularly useful for practical applications, as there are simpler ways to determine these features without using MRI scans. But however, from a scientific standpoint, it can be quite interesting to explore how accurately we can determine someone's age just by examining their brain. Moreover, these techniques can be applied to more practical tasks, such as predicting the progression of dementia. The choice of this task was somewhat pragmatic. There is publicly available large dataset named the IXI dataset, which includes 600 healthy subjects with varying characteristics like sex, age, height, ethnicity, and so on. Generally, most medical datasets aren't publicly accessible, and you usually need to request such data with a detailed scientific plan, hypothesis, and so on. Additionally, it takes time for these requests to be examined and approved. So we'll be using this IXI dataset. Let's move on to the basics of working with MRI data. I won't delve into the details here, but it's essential to understand some basic terminology. MRI scans are 3D arrays consisting of voxels. Voxels, simplifying it, can be considered as pixels in 3D. They have 3 coordinates in 3D space, while pixels have only 2. Each voxel has its intensity value, analogous to pixel values in regular image data, or 3 values for colored images in some color space. In MRI data, we only have 1 intensity value for each voxel, similar to grayscale images. Slices are two-dimensional arrays, planes in one of the axes. These slices consist of pixels with intensity values, and I will demonstrate you a sample scan in FreeView, a tool that is a part of FastSurfer. FreeView is a GUI tool that allows you to examine scans in each axis. This is a FreeView window, and we can load a sample image from the original dataset. Here is a sample scan where we can observe 3 slices in different axes: sagittal, coronal, and axial. The 4th view is for navigating through the 3D scan, allowing us to choose interesting slices for a preview. Additionally, we have the capability to adjust brightness and contrast here. And moreover, here specific voxels coordinates are visible here. So we can navigate through scan. So let's move on to the next slide about MRI sequences. Oh, here we got another slide for axial, coronal, and sagittal axis of the scan of the body. And As mentioned earlier, I will provide more information about sequences, but as a machine learning specialist and not a radiologist or physicist, I won't delve into intricate details as this is a highly complicated matter. However, in this slide, we can observe the same slice of the same brain acquired in different MRI sequences. Different tissues exhibit varying intensities for each sequence, and these sequences serve various medical purposes. Some of them are valuable for tumor detection, while others aid doctors in diagnosing different types of dementia, often correlated with the shrinkage of specific anatomical structures in the brain. Different sequences offer distinct contrast between white and gray matter in the brain, and it's worth noting that MRI is not limited to brain scans alone. For our purposes, we will be focusing on T1 sequences, which are also frequently employed in the prediction of Alzheimer's disease. You can also experiment with other sequences available in IXI dataset. It may lead to interesting results. However, be aware that it is impossible to extract tabular data only from T2 scans. You will need both T1 and T2 scans. Nonetheless, it is possible to use T2 scans with a ConvNet-based method after performing preprocessing steps. And today, we'll be employing two different methods to use MRI scans with machine learning models. The first method involves classical machine learning models, such as decision trees, random forest, and so on. This is because our input data, in this case, consists of normal tabular data. Qualified radiologist or specialized software like FreeSurfer or FastSurfer can provide information about volumes, thicknesses, and areas of specific anatomical structures in the brain based on an MRI scan. This data can then be used for modeling with traditional machine learning algorithms. In the next slide, I will explain this in more detail. And then the second approach involves using the entire scan without measuring volumetric data. This method employs deep learning, especially 3D convolutional networks. We can input the entire scan into the network and train a model to discover these relationships between specific regions of the brain on its own. Both methods are widely used and researched, while with many papers based on this approach. One of the challenges I encountered during my work with MRI data was finding a proper way to preprocess the MRI data for the model training. I will demonstrate how to do this later and aim to explain why it is necessary and show you the results of each preprocessing step. Now, let's begin with the first approach, extracting tabular data about volumes of specific brain parts using FreeSurfer. FreeSurfer is a toolkit designed for preprocessing, analyzing, and visualizing human brain MRI scans. With FreeSurfer, we can extract volumetric data from the scan. Let's take a look at the sample statistics file generated using FreeSurfer. As you can see, it's a simple CSV-like file with additional descriptions on top. It includes anatomical structure names here, the number of voxels belonging to each structure, volume in cubic millimeters, mean intensity value in that particular structure, standard deviation of intensity, and minimal and maximal values. And in its pipeline, FreeSurfer employs several preprocessing steps on the raw image obtained from the scanner. We'll discuss these steps in more detail later. And after preprocessing, the brain is segmented and the segmentation mask is created. We can view the sample created mask using Preview. As we can see here, the segmentation mask specified which voxel belong to which anatomical structure. Statistics are then calculated based on this mask. Before segmentation, scans undergo a registration process to a normalized template. In the registration process, we ensure that every single voxel corresponds to, for example, 1 cubic millimeter of the brain. This allows us to calculate volumes of specific structures in the brain just by counting voxels belonging to a specific class. This way, the segmentation mask is transformed into a stats file, which we have seen before. Here we can see the coordinates of specific voxels and to which class they belong to. And here we have the structure names and assigned color. And by counting the voxels, we are extracting the tabular data about volumes of the specific anatomical regions in the brain. But the main challenge with using FastSurfer is the considerable processing time, up to 10 hours for the full pipeline for the single scan, not for the whole dataset, for the single scan. Given our time constraints, we have an alternative solution, which is FastSurfer, a tool that utilizes convnets and achieve the same results. Brain segmentation using FastSurfer takes about a minute, significantly faster than FreeSurfer. FastSurfer also saves files at various preprocessing steps, some of which align with those used in the second approach, which is using convolutional networks. Now that we understand how it works, so let's proceed to demonstrate how to actually use FastSurfer. In this repository, we have a module called scans preparation. Let's take a look at the Docker file. FastSurfer comes as an already Dockerized tool, so there's not much we need to do. I've added some additional software used for preprocessing steps in the convolutional network method. This added toolkit is called FSL, and is also widely used when working with MRI scans. Brain registration requires an atlas, so we copy it to the container here. Here is the file. We can now build the Docker image based on this Docker file, which may take a few minutes if it's the first time. Okay, we probably need a few minutes to download the images. And additionally, I've included a simple script here to batch process the entire IXI dataset. In the script, we apply the FastSurfer pipeline here, performing brain image masking, which is also called skull stripping, converting from MGZ format to Nifty, which is usable in Python using Nibabel library, and applying brain registration, and then applying brain's image denoising. This script ran inside the Docker container. We are copying it here inside the container. And the second script is called run FastSurfer py, and it is used to execute the entire process. Remember to change these three paths here. the output data path, the source data path, and the directory with the licensed txt file. If you filled out the FastSurfer registration form, the license file should be already in your inbox. Download it to an empty directory and pass the path here to this variable path, not directly to the license file, but to the directory with this license file. And after that, you can just run the script. Now the FastSurfer preprocessing FastSurfer is running. It is predicting the segmentation mask in each axis separately. As we can see here in the logs, the scan is registered, and for each voxel, it corresponds to exactly this volume of the brain. This is the resolution of our scan. Now we are waiting for FastSurfer to perform extraction of the tabular data for the one scan. And after that, we can stop the process and examine the results. So now we are waiting. Maybe you have any questions? Okay, we have one of the scans processed by FastSurfer. So we can examine the results. We have three directories here, stats, MRI, and scripts. The scripts directories contains the full FastSurfer log for this example. After processing, you can check if everything is in order here. It can also be used for debugging. And in the stats directory, there are two stats files. The first one, aseg, I've shown you before. And the second one contains additional data on cerebellum segmentation. While I did not use this data in our tutorial, you can explore and add them to compare the results and assess their usefulness. You will only need to modify the stats file parser, which I will show you later. If you receive interesting results or encounter any problems reproducing this tutorial, feel free to email me. My email address is in the README of this repository here. After this, we can proceed with running the classification of these examples. First, we need to combine all the results into one large data frame with one scan or subject per row. I've prepared a parser in a module called XGBoost modeling, and the parser is called stats2CSV. This script finds and parses all the stats file in the specified directory. Comments are removed and the data is transposed so that every stats file is represented as a single line in our data frame. Then demographic data is joined. Well, this tutorial will be available later. It's kind of hard to follow live. I don't know. You have presentation available and the whole repository. I don't know if records of this tutorial will be available. This is a question for our hosts from Data Science Conference.
I'm pretty sure that the video will be available later. I think YouTube, but we'll ask.
Okay, thank you.
You're welcome.
So we can continue. We're running the stats2CSV file script. So comments are removed and the data is transposed so that every stats file is represented as single line. Then demographic data is joined. The key to joining scans with demographic data is called IXI-ID, which is used in scan file names and in the demographic data spreadsheet. So let's take a look at this file. As you can see, we have a few columns here containing demographic data. We'll be using only age, sex, and IXI ID to match the scans with this demographic information. Now let's run the script and transform several individual stats files into one data frame. Which we'll use for prediction. Remember to set these three paths here. The first variable is for the path to the preprocessed data set. The second is for the file with demographic data. And the third is for the output. So now we received this file. We can preview it. So as mentioned earlier, there is a single line in the data frame for every scan. So we transform our MRI brain scan into a single row in this data frame. And the last columns are for demographic data and subjects ID. So now we can proceed with classification using XGBoost. So in this notebook, we'll classify data prepared in the previous steps using XGBoost. First, let's import all necessary libraries. If you encounter any errors here, you need to install missing libraries from the requirements file. using pip install r requirements txt. This way, but I will not run this. So next, we perform data preparation step. We define the columns we'll be predicting, denote as Y columns. Then we specify columns to drop, which are not relevant to our task. After that, we load the data and split it into train and validation subsets. In our case, with only one scan per subject, this simple method suffices. Otherwise, you should split your dataset subject-wise. Scans from a single subject should belong to only one subset. Otherwise, it is considered data leakage. I've read many papers with this error, even in scientific journals with an impact factor. So be careful, because we don't want to produce such a poor science or projects. We then use the standard scaler to scale our input data. And the following step is data exploration, which is brief and limited, as it isn't very relevant to the tutorial's topic. Here we include the age histogram and the sex proportions in the dataset. Moving on, we proceed with classification. We use the XGB classifier to predict a subject's sex based on the volumes of the subject's brain. We encode the ground truth using one-hot encoding with a label encoder tool, initialize and train the model. And after that, we make predictions and calculate metrics on the validation subset. Because the validation dataset isn't used for setting hyperparameters, we can treat it as a test subset. We use accuracy and balanced accuracy metric. What about encoding other features? We are scaling our features using standard scaler, and all of the features which are in our data frame, I will show you. We don't have any other categorical features here in the data set, because all of our features here are numeric, not categorical. So we don't need to encode them. Only the sex here is binary. Okay, you're welcome. So let's proceed. We use accuracy and balanced accuracy metrics, recognizing that our classes are somewhat unbalanced. The results aren't really impressive, but they surpass those of a random classifier, of course. Steps for improving these results could include acquiring more data or performing hyperparameter optimization. Additionally, removing outliers from the data may be worth exploring. As seen in the age histogram, there aren't many examples for subjects older than 70 years. And however, achieving the best results wasn't the goal of this tutorial, but rather to demonstrate methods. And now we can train the model for age prediction, a typical regression task. We use the XGB regressor and evaluate results using mean squared error, mean average error, and R2 metrics. Based on the mean average error metrics, results appear promising. The R2 metric, which may be unfamiliar to some, is useful for quick assessment of regression results. It ranges from 1 for perfect regression to minus infinity. We can also directly preview the predictions correlated with grand true values. As seen, the predictions are accurate. It's worth noting that the aging process varies among individuals, is dependent on environmental conditions and lifestyle, making it a generally challenging task. At the left, we have our predictions, and at the right, we have grand truth. And as an additional feature, I've included a basic explanation of XGBoost model explainability. In this section, we explore the feature importance of the given models. The presented tables highlight the names of the features and their respective importance values. These values are derived from how frequently a specific feature is utilized in the model, which is composed of numerous shallow decision trees. While I won't go into intricate details of XGBoost architecture here, it's noteworthy that we can compare these results with state-of-the-art medical knowledge to validate the accuracy of our models. For example, from my personal and professional, of course, research, we know that fourth ventricle here is not correlated with subject sex from our medical knowledge. But for example, the amygdala and hippocampus are well-known predictors of subject sex. This may be a result of overfitting, or we need to acquire more data. But furthermore, from a scientific research perspective, this approach has the potential to uncover new biomarkers for various diseases this way. Maybe do you have any questions? So if no, we can proceed with the second method. Okay, so before we'll be feeding our convolutional networks with MRI brain scans, we need to pre-process them. And to explain all the necessary pre-processing steps, I have prepared a notebook with visualization of scans before and after performing specific operations. I will not even run these cells because it's only for previews. Here you can see the 10 random scans from the IXI dataset before pre-processing. The first observation is that the scans not only show the brains, but the entire head, including all surrounding tissues. This is unnecessary noise that we should remove following the well-known rule in data science called garbage in, garbage out. We are interested only in brains, as predicting age and sex will probably even be likely be even easier using images of the skull and face and this tissue here, the neck. The second aspect is the intensity values of voxels. We can observe the mean and maximal intensity values per single scan using box plots. As seen, the median mean value per scan falls between 1 and 200. These box plots were generated by measuring mean value of the scan for every scan and maximal value for every scan of the intensity of the voxels. So median of the mean values is between somewhere between 0 and 200, 100 and between 100 and 200. And there also are numerous outliers, which are even higher than 1000 mean value of intensity. And the same trend applies to maximal values. Some scans have a maximal value near 10,000 and even 20,000, while the median maximum value is somewhere between 1 and 2,000. Simple scaling methods are not applicable here. Dividing all the values by the maximal value or by the maximal value in the dataset simply won't work. I've tested it, and prediction using these methods are completely random. The model won't learn anything this way. And why is that? Intensity values are context dependent, as we have many different MRI scanners that work somewhat differently. And additionally, MRI data sometimes contains artifacts such as voxel with high intensity or so-called spikes. The proper way to normalize the scans to values between 0 and 1 is the histogram intensity rescaling. How does it work? We group intensity values into bins, similar to how a histogram works. We also discard or clip the highest and lowest values, those higher than 99.5 percentile and lower than the half percentile. Then we reassign voxel intensity values with indices, like index of the histogram bins. So once again, we are grouping values into the bins, like in histogram, and then we're reassigning the intensity values of the voxels with the indices of the bins in the histogram. This implementation is borrowed from this repository, which I suggest you to check out. This is mentioned here in the docstring. And in these plots, we observe mean and maximal values after applying this function. There aren't many outliers here. And in all of the cases, the maximal value equals 255, similar to normal 8-bit images. And the next step is skull stripping, which involves masking all the tissue that is not the brain. FastSurfer itself performs this task. If you prefer not to use the FastSurfer or FastSurfer, alternatives such as BET or XGBoost tools are available. The BET The BET is a part of the FSL, which we included in our Docker file for scan preparations. And the HDBET is a tool based on convolutional networks, and it works faster and sometimes even is more accurate. So to perform skull stripping in the other way than using FastSurfer, you can use XGBoost, which I used in my project for my master's thesis. but it will also need a GPU. In the plot, you can see a slice before and after skull stripping. Additionally, a mask is visible in the third image. Of course, those are only the slices of the scan and the mask. Those are 3D arrays, and we are only previewing slices here. And the registration process, which is recommended but not necessary, is the registration of the scans. We can see two rows of slices in axial axis. The upper row displays slices of scans before registration, while the bottom row is for registered scans. All scans present the same slice with index 90. In the upper row, for each scan, completely different anatomical structures are visible. So if you just take a look at this scan and this scans, this is just at top of the head, and this slice is from somewhere in the middle of the brain. So the essence of the registration process is to align the scan with a template called an atlas. The goal is for specific voxel in the scan and in the whole data set with specific coordinates to represent the same anatomical structure in the brain. How does it work? The registration algorithm finds characteristic points in a scan based on anatomical structure. Then it is transformed to match the atlas as closely as possible. You can define which operations are allowed in the matching process. This is called degrees of freedom. We're using 6 degrees of freedom, which includes translation in free access, which translation is simply moving if you are not familiar with the term, and rotation in free access. Other possible degrees of freedom involve affine transformation, but this can lead to the distortion of the scans. Although they may better match the template, they can result in distort results. We are using here in the Docker file, in the convert.ixi file, the 6 degrees of freedom using FLIRT, which is a part of the FSL toolkit. We're using only 6 degrees of freedom. And I can show you the MNI 152 atlas, which we are using in our pipeline. This atlas is based on 152 unbiased T1 MRI scans of brains from people of different demographic to create a mean brain image. The most mean image, the most standard brain as possible. Each voxel here, the atlas is normalized, and each of the voxels corresponds strictly to 1 cubic millimeter of the brain. It's even in its name, in the name of the atlas. So in the bottom row, we see the 90th slice in axial axis, and the presented anatomical structures are very close between different scans. And in this plot, we can see mean images before and after registration in specific slice. In the mean registered images, we can clearly discern specific anatomical structure. For example, here we can see ventricles. The left image is more blurry and illegible before registration. And of course, brains differ from each other, and it's normal for the mean image to be somewhat blurred. But as we see, registration definitely works. There are more closely to each other. Mean image, if you are not familiar with mean images, is just calculated by adding all the images and then dividing them by the number of images. Just arithmetical mean for each each pixel in this case separately. So we can see how registration works here and why is it useful. From a modeling perspective, using convolutional networks, registration is necessary mostly to remove the margins here, because those pixels, those voxels are redundant, are not useful for our task. And by removing them, by cropping the margins, we can simplify and faster our training process. And also I observed cropping the margins improves accuracy of the classification. Also, we can apply affine augmentations in more controlled way. Registration probably also helps models to better generalize between subjects. The next step is for bias correction, bias field correction, which removes artifacts from MRI machines to ensure stable contrast on whole brain scans by reducing intensity variations. It's just ensuring the contrast is stable on the whole scan. Here you can see scans before and after bias field correction and denoising. The last step, which is optional, is simply denoising images. You probably can see the noise in the image. And coordinates are generally invariant to noise, so it's not really necessary, but some of the images are more noisy, and this preprocessing step can fix it. So let's move to the next slide. I hope you are not bored, but we need to delve more into theory. To classify MRI images using convolutional networks, which are 3D arrays, we must use 3D convolutional networks. This is necessary to capture all the relations between voxels, not only spatial relations like space on a single slice, but also relations between slices. 3D networks uses 3D kernels to produce 3D feature maps. Similar techniques are used to classify videos to capture not only the spatial relations on a single frame, but also temporal relations, temporal which involve time. For example, if you want to classify whether a car is moving forward or backward, you need information not only visible on separate frames, but also the temporal relations between them, the time relations. This is analogous to MRI data. We want the model to capture all the relations between voxels, not just those present on a single slice. However, the challenge with 3D convnets is they are time and memory consuming. You will likely need a top-tier NVIDIA GPU to train a model with a batch higher than 1. So to address this problem, Pseudo-3D CNN models were created. In the publication titled Learning Spatiotemporal Representation with Pseudo-3D Residual Networks, authors present a less memory and less time consuming approach to modeling 3D data. Instead of performing one 3D convolution, the 3D convolution operations are split into two different two-dimensional operations. Two-dimensional operations. If you're familiar with the mobile net architecture for two-dimensional data, it can be easily understood. It uses almost the same technique. First, we perform two-dimensional convolution on each slice, and then we perform two-dimensional convolutions in the temporal axis on the produced feature maps. You can see three variants of pseudo-3D blocks proposed by researchers. They differ in the order of performing spatial and temporal convolution. They can be done in parallel or one after other, or both, parallel and one after other, as in the C block case. All of the proposed blocks use shortcut connection known from ResNets to avoid the vanishing gradient problem. As I mentioned before, I implemented these networks based on this paper. The implementation is linked as submodule in our tutorials repo. Here. Additionally, I've prepared visualization of building blocks based on my implementation, because these ones here are significantly simplified, and the visualization of the whole network will be using to classify the scans. I implemented in these networks on my own because I didn't find a satisfying implementations on a GitHub. So let's take a look at the building block graph. Okay, this is a visualization of pseudo-3D block building block type A. After every convolution, we use the ReLU activation function and batch normalization layer. Additionally, we use a convolutional layer for the ResNet shortcut connection to match the dimensions of the input and output feature maps. Then we add the features, just simply adding, and apply the activation function. And let's take a look at our models graph, where this block is presented as a single layer here. This is simplified. Here we can see the graph of the whole model. We use a 4 pseudo-3D blocks type A, followed by the max pool 3D. And these blocks, convolutional blocks, are followed by adaptive max pooling, flatten, and dropout layers. Then we have two outputs, one for age prediction, and which is regression task, and the second for sex prediction, which is classification task. To train the model with two outputs, we calculate two separate loss functions, balance them to have values in similar ranges, and they simply add them. Afterward, we perform normal backpropagation as for a simple single output model. If this is clear, we can proceed with training. One more thing. To visualize networks in PyTorch like that, to produce these graphs, you can use a torch view library, which is really useful for debugging and presenting the architectures of the model made in PyTorch. So this is the last notebook in our tutorial. Here we'll be training pseudo-3D convolutional networks on the preprocessed IXI dataset. To reproduce these steps, you will need a GPU acceleration, because training it on CPU will take too much time. First, we need to import the libraries. Once again, if you encounter any errors, remember to install the requirements. So in the next cell, we provide paths to preprocessed scans directory. This is the path I provide you on the one of the first slide of the presentation. You need to download it and unpack it. And in the next, here we need to provide the path to the demographic data spreadsheet which I've shown you before. This is downloaded from the project site of the IXI dataset. Also set the batch size to match your GPU VRAM. Generally, the larger the batch size, the better the results and faster the training process, but we are limited with our hardware. So moving to the next step, we are splitting the datasets to subsets to train, test, and valid data subset. And loading the data using data loaders. Let's take a quick look on the IXI dataset class. This class derives from the Torch utils data dataset class. We define basic transforms here, cropping margins, as I said before, and downsampling the scan, just simply by resizing them. Downsampling here is used to speed up the training process, but it isn't really necessary. You can explore this on your own and disable this resizing, downscaling. For the train subset, we use simple augmentations, flip, affine transformations, which include scaling and rotation, blur, and random swap. I will show you sample scans with applied augmentations in the next step. For all these transformations, we use the Torch.io library, which is really useful for working with MRI scan. You can use it to perform some of the preprocessing steps, and Torch.io also offers some downloadable MRI datasets, including IXI. This is the project site for the Torch.io. For the transforms, if you are familiar with Albumentations library for augmentation of the image data, this will look familiar to you because we can compose the augmentation pipeline or preprocessing pipeline the same way as in Albumentations library. This library offers us normalization, rescaling intensity with histogram values, the same as I presented before. And also clipping. Clipping for here you can see the other medical data from CT scanners. Transformation, augmentation. Here you can see the random elastic deformation, which is very interesting. You can try it on your own to apply this for our project. Maybe it will produce even better results. And so on and so on. Then there are many augmentation methods, and also Torch.io provides other medical image data sets, including our IXI dataset, but also the other data sets with MRI data. You can try this on your own with your own projects. Okay, so moving forward, because this function is this class derives from this dataset, Torch dataset class. we need to define get item function. We load here scans using the NiBabel library. It allows us to load nifty files as NumPy arrays. Here you have the name of the library to work with MRI scans with Python. After loading the scan, we rescale it to a range between 0 and 1 and load the corresponding ground truth. Those are already preprocessed scans with histogram intensity rescaling applied, so we only need to scale it between 0 and 1 range. Okay. So we are also loading the corresponding ground truth. Here is we are returning the image, age, and sex as a tuple, and those other functions are for matching the MRI scans with our demographic data, with paths to the corresponding scans. Also, we need to define this len function to get number of batches and optionally to shuffle a data frame. So proceeding with this training notebook, we are loading the data. And here is the visualization of brains after applying augmentations defined before. Now we are proceeding to train the model. We load the architecture defined before here. We are starting from random weights. And I've shown you before the visualization of building blocks and model architectures before. Here is the implementation. Here using pseudo-3D blocks and the head flattened dropouts and 2 outputs. And in the next cell, we calculate class weights to weight samples during training, dealing with this slight class imbalance. And in the next cell, we define the optimizer, the loss function for both of the outputs. We use cross entropy loss for the classification to predict text of the subject, and root mean square error as a loss function for the regression task for the age prediction. The training loop is quite normal. The interesting thing here is using 2 loss functions for two outputs. We multiply the cross entropy loss by 5 to balance the values of the 2 different loss functions. We can already start this training process. So we are using 2 loss function, and we are multiplying the cross entropy loss to balance these functions to them to be in similar ranges to train the 2 outputs proportionally. And as you can see here, they are still kind of unbalanced because root mean square error is significantly higher than cross entropy at the start of the training, but RMSE values will be lower in the next epoch, so it will balance itself on its own. Then we add the loss values here to train both of the outputs. After every step of the training, after every batch of the training, we calculate accuracy, balanced accuracy, and R2 metrics. Also, we evaluate the model on the validation dataset after every epoch. We also save the model based on the sum of balanced accuracy and R2 metrics. Here is the save best call to the save best model metrics. This is a simple class which remembers the last best validation metrics, and it's saving our model in the case if in current epoch we have better results than in the previous epoch. So as you can see here, the training process is already running. And the metrics on the validation subset are still improving after the first epoch. We don't have to wait. We don't have to wait for the training process to end because I've done it before. Here are the metrics evaluated on the test subset. We've achieved similar results compared to the previous method, maybe slightly better, but we are using 3 subsets here, and before in the tabular method we used only two subsets. We have about 0.8 balanced accuracy on the classification task and nearly 7 years of mean average error. In my opinion, those are satisfying results. If you achieve better results during your experiments, please email me about your method. I'll be glad to hear about your results and if you find this tutorial interesting. In the next cell, we have correlated ground truth and predictions for each of the subjects from the test set. And as you can see, the model is predicting each of the subjects with really high accuracy. We are really close here. So you can preview it on your own because it's really interesting. And then in the next cell, we have correlated ground truth and predicted sex for each subject, but we have the metric. It's better to preview that. And we can see the model is still converging really fast. Here it's the sum of the both metrics for R2 and balanced accuracy, and it's epoch after epoch. It's better and better. So we don't need to wait for the training to end. And as a bonus, I've prepared some suggestions for your own projects based on the knowledge from this tutorial. Firstly, you can work on the explainability of predictions given by the model. In medical applications, it's crucial for predictions to be explainable. We need to know the reasons why the model gives a particular predictions. If you're interested in this, you can check out my other repository for Alzheimer's disease prediction. I will send you a link here using Zoom. And there you can find the basic of explainability of the predictions of both methods, but those are just really the basics. It needs further work. Definitely it needs more work. But additionally, if you gain access to datasets like ADNI, BioCard, or OASIS, I can write the names here. If you gain access to these data sets, you can apply the techniques presented in this tutorial and use this repository. I've copy and pasted the link here. Another idea, which involves tumor and or cancer segmentation, goes beyond the knowledge provided in this tutorial, because segmentation is a quite more complicated task than a simply classification. But however, it's a very interesting topic that is worth researching, and it can lead to really interesting results. Because there was already a cases where when a doctor didn't see something which machine learning model could see. So machine learning models are already saving people's lives using these segmentation techniques for tumors or cancers. Furthermore, you can explore using pseudo-3D networks for classifying videos. This is also a very interesting task, but it's really memory consuming and time consuming. You will need a good GPU. And my last proposition is the detection of mental disorders using MRI scans, which is also a very interesting task. This is becoming a more and more popular topic, which new publication every year. You can find also other MRI datasets at the Open Neuro site. I will paste you the link here. This is a free and open platform for validating and sharing datasets. Here you have 640 public MRI datasets. Some of them aren't really useful for machine learning applications, but some of them are really big, with containing many participants, and you can find an interesting topic for your own project. Also, the pet data are available. This also goes beyond our tutorial topic, but those PET scans are also a method to imagine the brain, and they're also used with machine learning classification. It's another topic worth researching. So in summary, I believe that employing machine learning techniques in the medical field has a great future. The vast amount of data we can collect is nearly impossible to analyze with traditional methods. This allows us to really change the world through data, significantly improving people's health and well-being. The ability to predict diseases or age based on MRI scans with minimal medical knowledge right on our personal computers is truly impressive, and it feels almost like a magic to me. I am truly delighted to have the opportunity to share this knowledge and spark your interest in using machine learning techniques with MRI data. Thank you for your attention. Feel free to reach out with any question related to this tutorial. Here is my email address, and once again, thank you very much.
Thank you so much. It was very interesting and educational, and thank you everybody for participating and watching the tutorial. I will send the provided links by the tutor again in the chat for everybody to see. And the next tech tutorial will be held at 3 o'clock in 45 minutes. So see you then. Bye.
Bye, thank you.